Welcome everybody to the fall NISS-Merck meetup. I want to speak quickly because we have three great presentations today. Just a few housekeeping things. Obviously, we're using Zoom software. Everybody's becoming pretty familiar with Zoom software these days. Your audio line has been put on mute, and that's obviously to keep background noise to a minimum. If you have a question for the panel, we will do questions at the end of all three presentations, or at the end of when all three are done. Please use the Q&A button. If you put questions in the chat, we may find them, but sometimes we don't. So the sure thing is to use the Q&A button. We will address as many questions as we can at the end, like I said. Feel free to use the chat to talk with the other attendees. Just a couple of announcements before we get started. Many of you have seen other NISS-Merck presentations and realize that NISS is a nonprofit research institute for statistics. It promotes statistical research through lots of activities, including postdocs, conferences, workshops, independent research, and the affiliates program. The NISS-Merck meetup really does come from the affiliates program. And so those of you who are a member of pharmaceutical companies, I encourage you to get your company to become an affiliate. One of the advantages of becoming an affiliate is some of the things that cost to other people are free to affiliates. And one of those is the Essential Data Science for, excuse me, for Business set of webinars that's going on right now. You can see the schedule in front of you. Like so many things, all the details are at the NISS website, www.niss.org. A couple other events that are coming up: the Ingrid Oken Forum, Algorithmic Fairness and Social Justice, coming up in just about 9 days. You can see the presenters there. Again, information at the NISS at the NISS website. And also next month, in just about 1 month from now, NISS has been very active in the p-value discussions, and we decided to do something a little different, to have the statistical debates. Essentially, we've gathered a number of prominent statisticians, and they're going to talk to specific questions about p-values, confidence intervals, Bayes factors, and reproducibility. And you can see the exact questions and who the speakers are at the NISS website. Of course, today, what we're doing is the NISS-Merck meetup, and I think we have a particularly topical or timely topic, is what I was trying to say, in vaccine development. Certainly, the pandemic has affected everyone's life. Everyone is wondering when life is going to go back to normal. No one knows exactly how this is going to resolve, but certainly one thing that would help is a vaccine. Yet a lot of people don't really understand what goes into vaccine development. So we have three great experts today to talk about what really goes into vaccine development, and in particular, some of the things going into vaccine development for COVID. Here's the lineup. We first have Ivan Chan from AbbVie, who's going to talk about opportunities and challenges in the vaccine development. Josh Chen from Sanofi is going to talk about some considerations on developing a COVID-19 vaccine. And Mark Lipsitch from Harvard is going to talk about from testing to distribution, the importance of and challenges to estimating the protective effects of vaccines. So with that, I'm going to unshare my screen. Try to anyway.
At the top.
Yeah. Share screen. I think, am I unshared now, Jim?
You are, yes. And
Okay.
Ivan can share his while you introduce them.
Yeah, so let me. So Ivan is Vice President, Global Head of Statistical Sciences at AbbVie. He has extensive experience in discovery, biomarker research, early and late clinical development, and global medical affairs. He's worked in a wide range of therapeutic areas, including oncology, immunology, infectious diseases, neurology, women's health, biologics, and vaccines. Earned his PhD in biostatistics from University of Minnesota. He's a fellow of both the American Statistical Association and the Society for Clinical Trials. He serves as executive director of the International Society for Biopharmaceutical Statistics and has over 80 publications in statistical and clinical journals. So we're in very good hands with Ivan. Take it away.
Me okay? Yeah. Definitely, I'm really honored to be sort of invited to share a little bit some of my thoughts and experiences in vaccine development. And a lot of this experience that I have really is acquired when I work at Merck as my ex-colleague with Dan, but also with Josh, who you're going to listen a little bit more closely about some of the development that Sanofi is taking on COVID vaccines. So I thought I would sort of start the session by maybe sharing a little bit of the general considerations and some of the challenges and opportunities as we see it in vaccine development. And so this is like just a disclosure about my presentation. So many of you may have been working in vaccines, but many others are not working in vaccines. And there's always a question: what is the difference of vaccines compared to drug development, or the more basic questions of what are vaccines, right? And as we noted, vaccines a lot of times are biological products, and it was made from antigen or some attenuated virus. And the purpose really is to trigger the immune response for disease protection. And most of the times, the vaccines are either administered as a single shot or maybe come with a potential booster dose sometime later. And vaccines are typically for prophylaxis, not for treatment. And if you have seen some of the communications from CDC in the past, you would have noticed that vaccines has been cited as one of the greatest public health achievement in the 20th century. In fact, it's probably the most impactful public health interventions. And for example, the smallpox was eradicated in 1980 from the world by introduction of the vaccination. And for polio in the US is also elimination through the vaccination. As you know, the polio vaccines was started, development was started back in the 1950s. And so in the US, we are able to eliminate the polio at all. And there's a current initiative from the WHO to eradicate the polio from the world. And I think the Gates Foundation is working very closely with WHO on that initiative. So what are the benefits of vaccines? You could think about that from two different angles. One is the direct benefits, sort of like taking a drug. If you receive a vaccine, you would expect that you would be protected from the vaccination against the targeted disease, right? Like when we are taking the flu vaccines, our hope is that we would not get the flu during the winter time. And how you measure that, usually you could do that through the clinical trials, is more like a direct measurement, and it's more at the, if you look at the risk and benefit, it's at the individual level. And this is very similar to how we evaluate drugs. And however, in vaccines, there's also an indirect benefit to the society. And usually you may have heard recently about the term called herd immunity. Really is the protection of non-immune individuals by reducing the exposure and transmission of disease in the community. And this has had a great public health impact. And in fact, I think a lot of those benefits of the vaccination really come from the direct, indirect benefits. Let me just give you an example. This is one paper published by CDC colleagues. back in 2013 regarding the HPV vaccination program and how effective it is. So in this particular study, they reported the vaccine coverage at the time in the United States of about 34%, meaning only about 34% of the eligible population received the vaccine. And the vaccine effectiveness among those who are vaccinated is about 82%. So this is actually a very high effectiveness. And if you look at the clinical trial efficacy, actually much higher, but so this effectiveness is more in the real world setting. However, you can also notice, they also noticed that by this program with vaccine coverage about 34%, actually in the community, there's a reduction about 56% of the incidence rate. So what that means that there's actually a lot of herd immunity through this vaccination program. And notice there's only about 34% of people who are eligible to be vaccinated by this HPV vaccine actually receive the vaccine. So the herd immunity is a pretty simple calculation. You could do that, and it boils down to about 43%. So imagine if the vaccine coverage is a little bit higher to 50 or 60%, then the herd immunity could actually continue to increase. So that's the power of a massive vaccination and the indirect effect of the benefit. So I'd like to just take a moment to look at the different types of immunity that vaccine is supposed to induce, and in turn is going to induce the protection. The first type is called the antibody-mediated immunity or humoral immunity, and it's different kinds of B cells. Plasma cells is part of B cell, and immunoglobulins and different types. For example, IgG dominate about 80% of the immunoglobulins that we have in our human body. So the antibody titers after supposed to, say if you have the vaccination, supposed to increase gradually to a plateau at some point after vaccination and then start to decline. And so the function of these antibodies have a different functions and depends on the type of antibody. We've heard about the terms of the neutralizing antibody, right? And when we heard about the development of COVID-19, we wanted to make sure the vaccine can actually deliver the neutralizing antibody that they could bind the antigens and prevent or clear the first infection. So this is more focusing on the prevention part of it with the antibody. There's also a different type of T-cell immunity that can be generated by vaccination, and I'll call the cell-mediated immunity, but cytokines or T helper cells. In fact, the T helper cells are the ones who can stimulate the B cell production. And so in turn, it sort of generate the factory to massively produce the antibodies. There's also a T suppressors, regulatory cells that help regulate and control the level or quality of immune responses, for example, like the CD4. And thirdly is the cytokine T cells. It actually could recognize and destroy those infected cells. And this is where if the vaccine cannot prevent you outright, maybe if they have T cell responses, they actually, once the body is infected, they can actually destroy the infected cells, and that in terms of protecting from a severe disease. And there's different examples of the vaccines currently available. For example, the herpes zoster vaccines is thought to provide the kind of protection through T cell immunity. So it could really reduce the substantial burden once you get herpes zoster. And many of the pediatric vaccines are really the mechanism is through the antibody. So it's trying to prevent the children from getting the disease that are like the chickenpox vaccines, the HPV vaccines, for example. So, so these are the two basic kinds of immunities that the body reacts to the vaccine. In the next few slides, I would like to talk about a little bit on how we evaluate the vaccines in terms of safety, efficacy, and immunogenicity. With safety, obviously, we would assess the local injection site and systemic AEs, right, for a period of time, depending on the type of vaccine, the mechanism of action. And in general, we need a pretty large database just because we may be giving the vaccines to healthy subjects, and we are also giving them in a large volume, and so it would be millions of people would be receiving the vaccine. So the safety hurdle, sort of a safety bar, is very high from a regulatory standpoint. Just to give you an example, in the rotavirus vaccine development for RotaTeq, which is from Merck, and Rotarix from GSK, there's a previous vaccine by Pfizer who actually was associated with a safety called interception with the rotavirus vaccine. So as the second generation of vaccines are being developed, we wanted to really make sure this second generation of vaccine do not have the same problem with the safety. So a large clinical trial was actually conducted and recruited up to 75,000 children was enrolled in a phase 3 trial just to make sure there's no safety signal that we can see pre-licensure. And obviously, after the vaccine is licensed, there's a different mechanisms of post-licensure surveillance of the safety signals, either by CDC or by FDA, but also by the sponsors. There's a multiple network that was used to monitor the safety signal post-licensure. In terms of efficacy assessment, the measure of the relative reduction of disease incidence is the primary measure for vaccine efficacy, and it's a very simple formula. It's 1 minus the relative risk, right? But one of the really unique feature of the vaccine efficacy evaluation is that from a regulatory standpoint, because the vaccine is going to be given to a large number of people, that requires a really high level of evidence and precision. And what that translates into a statistical criteria is that we typically require showing an efficacy lower bound much greater than 0. Right? In drugs, if you show that, okay, the new drug is better than placebo, then that means as long as you can show a lower bound greater than 0, right, in terms of your efficacy measures, then it would be fine. But in vaccines, that usually won't work, and the regulatory requirement usually require a substantial lower bound, for example, anywhere between 20% to 50%. And I will get into that a little bit in the next slide. And so because of that, we usually need quite a large study, and especially if the disease incidence rate are pretty low. And because of the uncertainty of event rates, many of the efficacy trials are designed with the event-driven type of a design. So in other words, you'll be looking at the total number of events and then do a conditional on the total number of events, and you look at the distribution of the events in your vaccine group compared to your placebo group, and to make inference on that. Now, that would be just showing the maybe relative short-term vaccine efficacy. Another very important question that we need to answer for a new vaccine is how long the durability of the vaccine protection will last, right? Do I need a booster? And if I need it, when do I need it? So those are the questions we need either a clinical trial to answer, or we need a post-marketing sort of a study to continue more like the real-world evidence study to collect additional information. And in many cases, we may not have a placebo because it's in those kind of real-world studies, because once the vaccine is licensed, it's really unethical to continue to use placebo post-licensure. So historical control may be used, or when we look at, sometimes we also look at the population event rates before and after the introduction of vaccines to make those kind of adjustments. So this is really when we think about how we could use the real-world evidence, the vaccine development has been used for a long, long time. And this slide goes to show you a couple examples, right? How the impact of the vaccine lower bound requirement on the size of the study. And I took this table is sort of drafted based on, say, you were looking at a disease with about 5 per 1,000 instance rate, and we usually may target a phase 3 trial with 90% power, and assuming we're going to detect a 60% true efficacy. And if we look at the lower bound, right, going from a 0 lower bound, which is right, if we are designing a drug trial, we might require 56 events total using a event-driven design. Or if we don't use an event-driven design, you could just look at what's the instance rate and then do the sample size calculation. It will be about 16,000 subjects trial. But as we increase the lower bound requirement, the sample size increase substantially. If we say I need a 30% efficacy lower bound, then you almost triple that sample size. In the real example, in this herpes zoster vaccine that was developed back in the 90s and the early 2000s, and the company Merck used a lower bound of 25% for that particular vaccine efficacy trial, and the sample size requirement is about 38,000 and needed about a follow-up time, it's about 5 years time to complete that efficacy trial. So it just goes to show you that because of the high bar for efficacy, the sample size requirement actually could go very high. The third dimension about the vaccine development is assessment of immunogenicity. And as we know, we always are going to look at either antibody or T cell responses to help us understand the mechanisms of action of the vaccines. And also, if we understand how the immune responses work, we might be able to come up with a so-called correlates of protection, or say in the drug trials, you may hear the word surrogate endpoint. So there's a very nice recent, not recent, but back in 2012, that Stanley Plotkin, which is a very famous immunologist, and Peter Gilbert from University of Washington, they come up, as you pointed, to redefine the correlated protection for the both to unify this terminology for the clinical and statistical community who are working in the vaccines. So a immune marker statistically correlated with vaccine efficacy, basically it's predictive of vaccine efficacy, and that can be called a correlated protection. And then they put down 2 different categories. One is called mechanistic. If the vaccine, the immune response is actually is the causal agent to protection. Or a COP could also be non-mechanistic. If the immune response predicts efficacy, but is not a causal protection, which is in many cases, sometimes when we look at antibody responses, they may be non-mechanistic correlated protection for the disease. That's a very good example with the antibody responses that we measure for herpes zoster. That was sort of demonstrate that it is a good non-mechanistic COP. And why do you want to do this kind of correlated protection? So there are multiple purposes, and we could utilize the endpoint for bridging studies, comparing maybe new versus old formulations, or comparing combination vaccines, and all the consistency of the lot manufacturing. I will get to that in the next slide. And just having a correlated protection could also support regulatory approval, right? So, for example, we are developing the 2nd generation vaccine or follow-on vaccines. We don't need to actually conduct efficacy trial if we have the correlated protection identified. And so that's the beauty of that, having a very important understanding of the immune mechanisms and also the correlated protection. I won't be able to go into very much detail, but we give you a flavor about the different kinds of correlated protection that have been done in the past. In a very nice and beautiful story about this, how the correlate protection was used to actually support the licensure of Prevnar 13. This is the second generation of the pneumococcal disease for children. And if people are interested, they use this title-specific method to justify the antibody responses as a correlate protection, and in turn use that argument to design the clinical trial to obtain the regulatory approval for Prevnar 13. And there's also a regression type in statistical modeling and how we could model the immune response and the disease risk and to estimate the protection for different levels of vaccine potencies. And you might, and many of you probably are also aware about this Prentice criteria that we get to use a lot in drug trials, for example, in HIV or cardiovascular trials, and that is relying on a concept called proportional treatment effect experiment. And this is definitely have been also used in the vaccine area as well. And a more recent development is the causal inference framework, where we really talk about using a causal inference framework to look at the immune responses and whether they are in the causal pathway. And then a good application of the antibody titers for the herpes zoster vaccine. If people are interested in those two references, a good source of paper to look at. Finally, I want to also look at the vaccine in terms of manufacturing. There's the variability and stability of the vaccine because of the biological products. And we really need to demonstrate that you could consistently manufacture the vaccines. And sometimes we may require a consistency lot study where you need to study 3 consecutive lots in a clinical setting and demonstrate that all the clinical responses from each of the lots are pretty consistent, and there's a specific requirement on that. And the other thing to worry about, and is also if you're working on the live virus vaccines, because the potency might lose over time, for example, the chickenpox vaccines, so you need to establish the range of potency for manufacturing and also the product shelf life. So you need to study the safety at a high potency, make sure there's no issue from that point of view, but also have to establish the efficacy at near expiry potency. So so for those who are really interested in the vaccine development, this is one of the key issues that we have to work with our analytical department and to and incorporate those concepts into the clinical trial to understand the vaccine profile. Okay, moving on, just to give you a very high-level concept about the regulatory pathways in general. For vaccine efficacy, there's a traditional approval pathway that's through efficacy trial. There's also accelerated approval pathway if we know of a surrogate endpoint or correlate protection that could reasonably predict the clinical benefit. We can certainly do that. And there's also an animal rule approval pathway, although it's rarely used. But if there's no other ways, for example, when we are working on the bioterror vaccines, anthrax vaccines, because you may not be able to give anthrax to people, right? And people have talked about using the animal rule approval pathway to secure the approval of the vaccines. More recently, as we heard so much about the COVID-19 vaccines, and we're definitely going to hear a lot more from Josh's talk. And FDA issued a guidance about the efficacy and also safety criteria. And it's very explicit. As you can see that we require a randomized placebo-controlled trial, and we require a demonstrate the vaccine to have observed efficacy of at least 50% with a lower bound of 30%. This is actually a dual criteria, not only a lower bound, you also want to see a observed efficacy of at least 50%. And then they also make a very interesting point about potential use of platform trial. As right, if we have multiple vaccine candidates that might be developed around the same time, actually a most efficient way might be to conduct a platform trial where you could have multiple vaccine and a single placebo can be used in that study. And they also talk about once the COVID-19 vaccine has been proven and has been approved, for second generation of the COVID-19 vaccine, you could actually use a non-inferiority trial design to establish the efficacy. So in other words, you don't need to conduct a placebo-controlled trial anymore. But that actually is also a pretty rigorous bar because of the vaccine is going to be efficacious. So it's how you determine the margin is a critical component that one has to consider and discuss with regulatory agencies. From a safety requirement point of view, actually, this is a pretty straightforward and is very similar to other preventive vaccines, right? About at least 3,000 individuals. And then you look at the safety, the local and systemic AEs. There's a special AE of interest called vaccine-associated enhanced respiratory. So, and this is for all those manufacturers who are working on a vaccine that we need to be diligent about that. The way I see it, there are a few opportunities that we could accelerate the vaccine development. One is because we now have a lot more knowledge about the genomic sort of knowledge on the disease. And also we could leverage the vaccines, immune marker and correlate protection. And there are also many areas of clinical trial innovation that one could learn and continue to on, for example, novel endpoints or using the likelihood ratio sequential methods or adaptive designs. And I know Josh and I work on a trial for the HPV vaccines, and that is a beautiful application of the adaptive designs. There's also population enrichment design that could be used. But I do think the platform trial and master protocol is a perfect storm and an opportunity for developing like COVID-19 vaccines or any of the potential vaccines, maybe in a outbreak setting where you have many of the sponsors developing it around the same time, that we could leverage those kind of things. And I want to draw the attention to a couple of the very recent development, how the acceleration of vaccine development was conducted. One is the Ebola vaccine. This is developed by really a private public partnership between Merck and NISS, WHO, and many other organizations. And it's really developed during the Ebola outbreaks in Africa. And phase 1 study was initiated in October 2014. And about 3 months later, the phase 3 dose is actually selected from this phase 1 study. And then 2 months later, the phase 3 study was initiated. And I was fortunate to be the lead statistician when I was at Merck at the time and working with WHO on this randomized clustering study design. And ultimately, the vaccine was approved in December 2019. So you see, it takes about 5 years. And this is actually a very good compared to usually it takes about 10 to 15 years in the past. But I think with the COVID-19 development, we might have a really good opportunity to accelerate that. And if you think about these timelines, right, the genome sequence of COVID-19 was identified in January. And in March, we started phase 1 study with the messenger RNA vaccines. And the study design, right now we have 3 phase 3 trials in clinical trial in the US. And it's really follows strictly the FDA guidance. And so it's a tremendous collaboration. We might actually be able to develop the COVID-19 vaccines in 12 to 18 months instead of years. So that's really really good for the public health and because of the collaboration of the private and public institutions. So just in summary, vaccines are very important medical products. They have great public health benefits. And there's a multitude of vaccine development challenges due to its biological variability. But I think we have a pretty good handle, and there's also multiple tools that we could use. And many of the opportunity exists for accelerating the vaccine development, both in terms of innovative trial designs, use of a biomarker or correlate protection, as well as private-public partnership. So And I'm going to stop here. I know there will be there are a couple of the list of references that people feel free to take a look from the slides. I won't go into detail. So Diana, maybe I hand back to you for now.
Yep. So Jim, if you could share Josh's slides. Thank you for that great, just laying out the framework. Because like I said, not everybody is very familiar with that. So you've set a nice base for everyone, but we need to move on. So our next speaker is Josh Chen. Josh received his PhD in statistics from the University of Wisconsin-Madison. He is currently the global head of biostatistics and programming at Sanofi Pasteur. Before joining there, Josh worked on clinical development of small molecules and biologics and vaccines at Merck. His experience spans many therapeutic areas with a major focus on human vaccines and antiviral drugs. He has extensive experience in study design, conduct, and reporting of international clinical trials, from proof of concept through regulatory approvals. His primary research interest has been in clinical trial design, including group sequential methods, adaptive designs, and multi-regional clinical trials. He was co-lead of the cross-industry MRCT group, the Consistency Working Group under Pharma, and the Society of Clinical Trials. He's a fellow of the American Statistical Association, one of the experts in this area. So I'll pass it to you, Josh, but before I do, let me remind folks that, because I think I forgot to say it before, that the slides and recording of this will be available at the Merck, or excuse me, at the NISS website sometime after the presentation. So with that, it's all yours, Josh.
All right, thanks, Dan. Thank you, and Jim and Glenn for organizing this session. I think this is a very good opportunity to share with our colleagues here. And also thanks to Ivan to set the context, the background for vaccine, make my life much easier. So to control this global COVID pandemic, I think we have two powerful tools. One is about responsible measures, actions, like for example, social distancing, wearing masks, washing your hands. And the second one, powerful tool, of course, that's vaccines. And hopefully what we are doing here, my colleagues Bo and Ya as part of this presentation, and those of you who are working in COVID vaccine development, you are the true heroes trying to bring the vaccines to control this pandemic. Next slide, please. It's just a typical disclaimer. Next slide. So 3 topics I just want to discuss with colleagues here. So one is what's the science behind the COVID, and what are the regulatory pathways which Ivan also touched upon, and of course, with all this science and the regulatory pathway, what would be the clinical plan and the design considerations. Next slide. So coronavirus, that's get its name. It's a family of viruses. It actually got names from the spikes. If you look at the picture, the beautiful spikes, the virus need those spikes to attach to human ACE2 receptor and get into the human cells. So the spikes are very important, and the spikes look like crown. So that's where the coronavirus comes from. And it's a big family of viruses. Before SARS, COVID-19 came into our side, we were living with coronaviruses. They cause common colds, maybe just a few cups of tea or a few nights of good sleep, you will be able to handle that. And typically year, it accounts for probably 15 to 30% of the cold, like flu season. They are harmless. So in that sense, they've been around for a very long time. And coronavirus came to our attention. Some of you probably noticed MERS, that's Middle East Respiratory Syndrome, and also SARS, that's about 20 years ago. And then now, of course, this is SARS-CoV-2, that's a virus causing COVID-19. COVID-19 is a disease. And sometimes we just use COVID-19 virus and disease. Next slide. So if you look at the three lethal coronaviruses, SARS, MERS, COVID-19, what's the difference between these lethal ones versus the harmless ones? People look at the genomes, they identified two major differences between the harmless one and the lethal ones. One is the spike protein. That's where the three clearly bold virus. And if they also have some difference in nucleocapsid, genomic difference. If you look at vaccines right now, most of the COVID vaccine actually target this spike, trying to prevent the virus, novel COVID-19, getting into human cells. Next slide. Hello? Sorry.
You see
Okay,
it?
thanks.
Yeah.
Yep. Okay, thank you, James. So there is a very interesting question, right? Because the common cold coronaviruses have been around for many, many years. Can it cause like cross protection, right? In fact, this is a very nice paper showing that indeed, yes, potentially those who were exposed to coronavirus, common cold coronaviruses before, they potentially could be protected in a certain way from the COVID-19. And if you look at this in left side, this plot. So what happened? Researchers they took samples from health donors. So that's a gray, 68 of them, and also take samples from the COVID patients who had developed some type of immune system in the immune system. 18 of them, the red bars, and then they compare, they challenge the cells extracted from there with COVID-19 spike proteins. And interestingly, if you look at the red, that's 85% had response, T-cell response, of course, because they exposed to COVID-19 before. So of course, the human immune system started reacting. But for those who never exposed to COVID-19 before, but they potentially exposed to the common cold coronaviruses before, 35 of them actually had response, had reaction. So this is actually a very interesting question, because when we design our efficacy trial, keep that in mind. This potentially have some implication about this cross protection, may affect our vaccine efficacy in certain ways. Next slide. As of August last month, there were about 200 vaccines candidates in COVID vaccine candidates in development. And this table shows the platforms, the categories of these vaccines. And for each platform, what, based on the data, maybe a lack of data, maybe some knowledge of the biology. So what would be expectation for the safety, for how fast you can go with this platform to develop COVID vaccine, and also logistic of global distribution. And again, also look at predict what the neutralizing antibody could be, because the neutralizing antibody, that's potentially, that's going to be the immune response to kill the virus. So the three categories with check marks are those at that time, there were clinical data, including the viral vector, so that's adenovirus. Some of those are in that category, leading candidates in that categories. And with mRNA, messenger RNA, for messenger RNA, I think one of the challenges, which is the feasibility of the logistic global distribution, because they were, you need to keep them stable. So you have to use a very low temperature. Some of those leading candidates in development require about -20 Fahrenheit, and there's another one -7. That's another requirement for messenger RNA. You need to look into that. And then the third category, the bottom, that's a protein. So that's a more established platform looking at the protein recombinant vaccines. Next slide. These are the leading candidates in clinical as of early August. So the data, I guess the timelines status probably already changed for some of the project. If in the mRNA, messenger RNA categories, these are the two leading candidates, and also protein-based. That's where our company actually is working on that. Viral vector, that's where adenovirus, measles, also considered by these companies. And if you look at this point, Q3, we have multiple candidates already in phase 3 development, including the two mRNA and also the viral vector from AstraZeneca, Oxford. Next slide. So the approach we take in Sanofi, we are looking at the two different platforms. The bottom one, number 2, that's the mRNA. So that's very similar to some of the leading candidates ongoing in phase 3. The first one is kind of unique. It's based on baculovirus recombinant vaccine approach. So that's where we use this insert the genome of the virus into the baculovirus and grow the protein, the virus in cell culture, and then we extract the protein with the virus combined with adjuvant, and that's a established approach, recombinant vaccine. This is and the other benefit of doing this is that because of the manufacturing capacity, that's also easily scale up, because in global pandemic, you need to give this to billions of people. So you need to manufacture, actually you need to be very easily scale up. So that's another benefit of doing this. Next slide. 2 points about this protein-based. One is there is a flu vaccine actually based on this platform, flu block. It's using the same platform to protect the flu. If you look at the Kaplan-Meier curve, that's actually reduce the flu incidence by 30%. On the right side, this platform was used before for SARS about 15 years ago. At the time, the SARS very similar to the novel coronavirus. And at that time, it didn't proceed beyond clinical. So that's a, but still some of the early work involved in this area. Next slide. So question, I now switch gear. Let's talk about how can we accelerate vaccine development for novel this pandemic. In traditional paradigm, that's on the top, that's where Ivan mentioned typical vaccine development will require 10 to 15 years from research all the way to licensure. Now, for pandemic, that's a bottom panel. Pandemic, so we want to develop this vaccine, COVID vaccine, in 12 to 18 months. So how can you reduce from 10 years down to 1 year? And the opportunities here, there's a very nice paper from New England Journal of Medicine. I adopted this picture. The potential opportunities here, one of course is the R&D part, right? So how we can get the vaccine licensed as soon as possible. And the second part is the manufacturing. You have to manufacture, make this vaccine available to billions of people. And that's a opportunity number 4, manufacturing, scale up. That part, I guess I'm not going to spend too much time on that, because that's really, you invest early on. You don't wait till you have a good data from phase 2, phase 3 to start scale up. You can start investment very, very early on. That's what people are doing now. So the focus for my talk here is truly about 1, 2, and 3. That's in R&D space. One, that is what is the regulatory pathway, right? So if you follow the traditional paradigm pathway, you have to get to phase 1, move to phase 2, move to phase 3. That's going to take 10 to 15 years. Now, is there any way we can accelerate, make this faster? Now, with this regulatory pathway established, then we can look at 2 and 3. In preclinical space, can we accelerate animal testing? Can we accelerate some of the early part, maybe in parallel to clinical development, in certain way to accelerate entering into clinical, right? Number three, that's where in clinical development, maybe we can go faster instead of having the traditional phase 1 stop, phase 2 stop, phase 3. Can we streamline, make this clinical development faster? So the opportunity truly, my talk focus here is 1 and 3, regulatory pathway and clinical design, clinical plan. Next slide. Well, this is from Nature. It's pretty much said, well, there's no accelerated pathway for COVID-19 at that time back in May. And for any other emerging infectious disease, no accelerated pathway. Next slide. I will also talk about this. On the left, these are some of the regulatory pathway in some other areas, some different settings. If you look at the 3 different way, emergency use authorization up to accelerated approval to regulatory approval. So all these are increased with more data. And the bottom one, emergency use authorization, sometimes people just say, well, I have very limited data. But based on the knowledge, based on some extrapolation, we believe it's going to work. Then we start using that and collect data while it's being used. Accelerated approval, that's where maybe biomarker can be used, surrogate endpoint can be used. Now, regulatory approval, regular approval, traditional approval, that's where all the efficacy 9-yard need to be reached. All efficacy data, long-term, and everything need to be in place before get approval. So if we look at in these options, some of you probably noticed aware Russia, the first approved COVID vaccine, there are people, many people, scientists, very worried, alarmed, because based on very limited data, pretty much just one phase 1 data with 70, 80 people with the clinical immunogenicity data and some limited safety data. And the Sinovac approval, that's approved for emergency use. That was based on phase 2 immunogenicity data, but no efficacy data. Now, I've also mentioned earlier, FDA guidance, which is the circle here, the green, issued in June. That's where they set the stage, how the COVID vaccine need to be, will go through the regulatory pathway. And I'm also going to talk a little bit about this EUA plus, emergency use authorization plus, which actually came out last week. Next slide. This is the FDA regulatory guidance issued in June. Ivan already talked about it. The only point I want to make is that it's the traditional approval. It's pretty much said, no, we need to go through the traditional approval paradigm. We cannot take the shortcut. So that's it. That's the bottom line here. The justification for that, no accepted surrogate, and also mentioned EUA possible. Next slide. This is interesting development because many of you probably are aware there's a discussion when the first COVID vaccine will be approved in the US. Someone mentioned end of October, someone mentioned end of this year. So all this depends on what data need to be in there, right? And what pathway should be. And this is where EUA Plus came out. EUA Plus, it's they use a EUA Plus because it was from which was used for some of the therapeutics in the past few months. So in the coming weeks, we probably will get more guidance on this EUA plus pathway. FDA is going to issue that. Next slide. Now, with the regulatory pathway in mind, now let's talk about what clinical development plan, right, we have, and how we can design our trials for phase 1, phase 2. Now, the phase 1, phase 2, this is where we can go faster, because typical phase 1, you take a pause and get into phase 2. Now here with phase 1/2, you will be able to look at very brief, short, sentinel cohort, very small cohort, look at the safety, make sure the safety is there. Then you can expand into phase 2. Dose formulation, pick the dose for phase 3. And the idea here, when we do this, we need to keep in mind what we need to put in for the dose formulation, right? Do we want to have 1 injection or 2 injections? Like in pandemic situation, ideally you want to have 1 injection, just 1 injection and protect it. But in most cases, you actually have to deal with 2 injections, because the human immune system need to get primed first, then boosted with the second dose. Second injection happen probably 1 month or 2 months after the first injection. And right now, most of the COVID vaccines in development, most of them are 2 injections, at least 2 injections. And there are some 1 injection in consideration, but I guess majority of those will require 2 injections. And the other point is, do we want to, in phase 1, we need to consider adjuvant for some of the platforms, because adjuvant, that's where to boost your immune response to the antigens. And that's going to be important in pandemic situation. Next slide. Now, with that in mind, we need to make a decision at the end of this phase 2. Dose, how we pick the dose. We need to look at biomarkers. Biomarkers, most important one, a lot of people looking at is the neutralizing antibody titers. That's where the antibody in the immune system is going to attack the virus, kill the virus. But also look at other immune response as well. And adjuvant, I just mentioned earlier, adjuvant is important in this case, because in pandemic, if you have with adjuvant, if you have similar immune response versus 25 microgram, that means you can, by adding adjuvant, you can increase your manufacturing capacity by 5-fold. So instead of giving those to 200 million people, you can give to 1 billion people. So this is important in the pandemic situation with the adjuvant. And of course, how you pick what's the criteria you pick the dose? Do you want to go with the minimum dose? That way you can manufacture-wise, you can give to more people, or maybe safety also probably going to be cleaner. Or you want to go to the highest one, because you don't know if the immune response going to stay there is enough to kill the virus. And how you compare that to convalescent samples. These are samples you collect from patients. So you know for patients, they have certain level that's probably compared to patients. How you want to compare to that? You want to be much higher? You want to be 3 times higher? And of course, when you consider that you have some at-risk population, like elderly, they tend to have lower immune response. So how can you give the dose to protect those higher elderly? And on the right side, these are the 2 publications which just say, one said, well, if you look at patients, COVID patients, after infection, after 3 months, well, their immune response goes away. And the other paper said, oh, no. 4 months later, they're still there. So there's a contradictory message. But all this means, what can we do, right? Can we do that, boost that, how to pick the dose? All right, so next slide. Randomization for phase 3. For phase 3, how you randomize? Here, this is, I guess, one of the discussion point. You do want to use 1-to-1 versus a 3-to-1, 2-to-1, unequal randomization. This is a just you can look at the reference. It pretty much says if you have 3 to 1 in this example, you actually increase the chance to see higher chance to see imbalance, which is critical for pandemic situation. You must have clean safety for vaccines. Next slide. Another point, some of the recent news, you have seen some of the sponsors start increasing their sample size for phase 3 because they want to have more diverse population, which this is important. Is in the guidance, they say, well, at risk population, you want to include those because they are intended to be used in this population. Like for those with some of the minorities, they tend to have the highest casualties in those. So when you want to use in those people, you want to include those people as well. And of course, the second bullet here for subpopulation, you cannot demonstrate efficacy within each individual population. That's where Ivan's point, they talk about correlate protection and all this biomarker extrapolation. All this will come to help. This is an opportunity for us to make a contribution. And of course, different population have different children, they tend to have lower risk. They also truly put safe vaccine for children. So for those, probably want to pick the established platform. Next slide. The other point is what kind of alpha spending function, right? Overall spending function in the bottom one, you tend to save all the alpha for the final analysis. Pocock, the top one, tend to spend more alpha the early part, so can stop early. So here you need to have a clear discussion about what's his speed and how much you want to save power for the final. Next slide. With all this science, with regulatory pathway, with the clinical development plan designs, now I also want to raise this point. This is the point here on the right bottom. It's truly speak to need to be adaptive. Need to be adaptive because we don't truly understand the virus. We don't truly understand the disease. And we don't know the epidemiology, where it's going. So with that in mind, what if his growth is not as fast as hoped? What can we do? What if the vaccine efficacy is not as high as expected? Then we'll give up. What if the approval or some of the early approvals, later you cannot use placebo anymore? What can you do? What if the second wave in the winter coming up going to be as bad as the 1918, this Spanish flu, second wave, the worst, killed most people. So all these uncertainties make us need to be more vigilant and adaptive. Next slide. I use this picture because you remember Forrest Gump trying to catch the shrimp, but a lot of shrimp in the ocean, but you don't know where to catch. And here you want to get the cases. You know a lot of things happening in this state, in that state, but you don't know where exactly. So you're trying to chase the cases. And you're trying to do this for 10, maybe 5, 10 phase 3 trials. And all of them should be successful. It's a very challenging situation here. Next slide. Now this is concluding remarks. Just want to reflect, just as I said, the science part, we're still learning. It's a steep learning curve, and now we're still learning. And of course, the race, we want to go develop this COVID vaccine, we need to be risk-free. That's the best, but we still want to go fast. And on this note, I want to reflect what Ivan mentioned earlier. This safety profile of the COVID vaccine must be safe, because it's going to be used for billions, millions, billions of people. So this safety is something we cannot sacrifice. And of course, we need to be more adaptive than just one size fits all. And the last point, which I think is truly true for statisticians, we will have great opportunities here to really make a difference. Now with that, back to you, Dan.
Thank you so much, Josh. There was just a lot of really important information in there. So we really appreciate your expertise and guiding us through that. I do note that we're a little behind time. We're in a global pandemic. and this is important information, so we will certainly go over. I don't want to rush Mark too much, and I'd like to introduce him now and get him started. Mark Lipsitch is a professor of epidemiology at Harvard School of Public Health, primary appointment in the Department of Epidemiology, a joint appointment in the Department of Immunology and Infectious Diseases. He also directs the Center for Communicable Disease Dynamics. His research concerns the effect of naturally acquired host immunity, vaccine-induced immunity, and other public health interventions on the population biology of pathogens and the consequences of changing pathogen populations for the human health condition. Some of his work is motivated by practical questions in public health, such as vaccine design and intervention targeting, and some is motivated by classical questions in population biology, such as how to explain patterns of coexistence of pathogenic strains in space and time. Mark received his bachelor's in philosophy from Yale, completed his doctoral work in zoology at Oxford University as a Rhodes Scholar, and did postdoctoral work at Emory University and at the CDC from 1995 to 1999. He is joined the faculty of Harvard School of Public Health in 1999, and we're very happy to have him with us today. So thank you, Mark.
Thank you very much, and thanks for the opportunity to be here. 1999 sounds like a long time ago. Actually, last week seems like a long time ago, as I think it does to many of us. Thanks to my two colleagues for setting the stage for many of the things that I'm going to talk about. And I'll just jump right in. Here are my disclosures, mostly unrelated to COVID and some unpaid advice that is related to COVID. You heard in the first talk that there are two ways to use vaccines. One is the direct protection approach of finding the people who most need the vaccine, who will potentially benefit the most, and vaccinating them directly. And on the other hand, the indirect protection approach, which in this particular epidemic seems to be vaccinating probably young adults who are the major transmitters, and thereby protecting the elderly and the others at high risk. That's quite weird, actually. This is perhaps almost unique in that usually we think of the kids as the reservoirs for most directly transmitted diseases, but this one, at least so far, seems to be, if anything, driven by young adults, but sort of everyone. And when we think about how to use vaccines, there are some requirements for using each of these approaches. The protect the vulnerable or direct protection approach requires that the vaccine be effective in those individuals who you're vaccinating. Otherwise, of course, it doesn't help much. And the challenge that I want to talk about in this talk is that it's unlikely that we will know whether the vaccine works specifically in the most at-risk subgroups at the first so-called successful interim analysis, or the first time we see an overall signal of efficacy, because by definition, if it's the first time, that means it's pretty close to the minimum sample size overall, and that will mean much less than that sample size in the vulnerable populations. So unless there's something surprising, it's likely that we will be uncertain about that, at least in the early phase. On the other hand, the indirect protection approach requires that the vaccine actually can produce indirect protection, that it reduces transmission either by preventing infection or get infected, but have been vaccinated. And again, it's unlikely that we will know this. Oh, my internet connection seems to be. It's unlikely that we will know this for most vaccines because the primary endpoint in almost, I think, all trials is symptomatic virus-confirmed disease, and only some of these are measuring infection or infectiousness proxies. So in this talk, I want to talk about three partial solutions to these problems, and some other partial solutions actually, but focus on three. based on work that we've been doing, some of it before COVID and some of it since. So the first approach is to measure the impact of a vaccine on infection using post-trial serology. So I mentioned that we are likely to know the primary endpoint is in most trials, if not all, symptomatic disease that's then confirmed to be caused by SARS-CoV-2. But even before COVID, we were beginning to think about the issue that you might want to measure infection as an endpoint, regardless of whether it's symptomatic. And this is a primary case in point. We didn't know about SARS-CoV-2 at the time, but when infection can lead to transmission, even if it's asymptomatic or subclinical, that's a prime reason why you might want to know that endpoint. To do that, what you need is the marker of infection that's distinguishable from vaccine immunity. And the simplest example of that is that because most vaccines are directed at the spike or S protein, antibodies to other components of the SARS-CoV-2 could be markers of infection rather than immunity, rather than vaccine immunity. You also need pre and, if possible, and at least post-study sera from a sample of or all participants. And the reason we started looking at this in the way that I'll describe in a second is that when you study an endpoint that involves symptoms, and there's also asymptomatic infection, a vaccine efficacy estimate against based on that is going to be biased toward the null when you have is going to be biased toward the null compared to the true efficacy against infection. And that was the framework we were thinking in a year and a half ago when we started doing this work. So I'll describe work that was reported in two papers, one in the American Journal of Epidemiology and then in a commentary in Nature Medicine during COVID by these collaborators led by Rebecca Kahn, shown here on the left. So so now this is going to be a little bit about the statistics of it and how we looked at the problem. So there's a bias in vaccine efficacy estimation when you consider only symptomatic infections. So if you have a randomized trial and you have people with vaccine and the control, some of those people will get symptoms, some vaccinated, some unvaccinated, perhaps fewer among the vaccinated if the vaccine is doing something. And then in the people who are not symptomatic, you don't know what happened to them. And so if the vaccine is preventing some infections, then in the vaccinated group, you will have fewer total infections. You will have, therefore, more people at risk in the vaccinated group. But if all you're observing is symptoms, then you perceive that the people at risk are all these people in pink down below. But actually, many of them are not. These individuals who are orange on the top row but pink on the bottom row are thought to be at risk but aren't. And because of that misclassification, one gets a inaccurate estimate of vaccine efficacy that biases the estimate toward the null. So what we did was to use a simulation model of an epidemic, and I won't go into the details of it because it's published in the American Journal of Epidemiology paper and for reasons of time. But essentially, we simulate an epidemic in a set of partially connected populations. We use this susceptible and exposed, infected, and recovered framework that used to have to explain before COVID, and now has been in the news so much that many of you at least will have seen this before. We run the epidemic with a vaccine, keeping track of people's true infection status, and then simulate a trial within that epidemic. And we assume that the vaccine is leaky, meaning that it reduces the probability of infection upon exposure. And so this summarizes what we find. We're looking at a vaccine that actually reduces susceptibility to infection in this simulation by 60%. And if you could observe every infection shown in the gray bar without any relying on symptoms at all, then you would, in fact, applying a Cox proportional hazards model, as is standardly done in these kinds of studies, you would get the right answer of a 60% efficacy estimate. If you only look at symptomatic cases, or look, let's forget approach 3 for brevity's sake. If you only look at symptomatic cases, as I said, you get an estimate that's biased toward the null. And then in various approaches that we describe in that paper, we find ways with either high effort, with multiple measurements of serologic status at the middle and end of the study, or even with a sample of individuals in the trial who are tested at the end of the study to see if they became infected, and then timing of infection in the unobserved ones is imputed, we can get a corrected estimate that's close to the right answer at relatively low cost. So, so one way that one can approach this problem of that we thought of it initially when we did the work as a problem of bias in vaccine efficacy, but is also a problem of just how do you detect infection when you're not following everybody all the time, is to do these tests of a sample or all of the participants of a trial at the end with some marker of infection that is distinguishable from vaccine, and that provides an unbiased estimate of the impact on infection, and that would help to provide evidence about the potential of a vaccine for herd immunity. The second approach, which I'll describe more briefly for reasons of time, is work that we did. Oops, I lost Rebecca. There she is. Rebecca's photo. She led this work as well with other collaborators. The second approach is to try to estimate infectiousness in an individually randomized vaccine trial. Traditionally, that doesn't work because we don't know who infected people in a vaccine trial. We only know whether someone got infected. But at least under idealized circumstances, and I don't know whether this will apply in practice to SARS-CoV-2 vaccine trials, but it's an approach that we were developing again before COVID began. It's at least in theory an approach that can help to solve the problem. And the idea is essentially, as the title of the preprint suggests, which should be out in medRxiv almost any time now, the idea is to use pathogen sequence data and contact tracing data to enhance a vaccine trial in an emerging epidemic. And so I'll just use this as a teaser and hope that those who are interested can have a look at the preprint. Essentially what we do is we simulate epidemics in a way that very much like I described in the previous example. And then at the same time, we simulate the evolution of the pathogen within the hosts and between the hosts. And what turns out to be the case is that if you have especially the deep pathogen sequence, meaning not only the most common variant, but in an RNA virus, there's variation within a host in the pathogen sequence. So if you have the deep sequence plus contact information, you can recover almost without bias an estimate of the infectiousness effect of the vaccine, which here is just assumed in our simulated data to be 30%. And we can get up to about a 28% estimate using contact tracing data as well as the deep and consensus sequence. And I apologize for doing this so briefly, but really I don't think this is the solution for this virus, but I think it is a new approach that could be used in vaccine trials in the future, where we incorporate some of the new kinds of data that we have. With Natalie Dean at the University of Florida, I've been thinking about some of the other approaches to get at both these infectiousness and herd immunity effects that I mentioned, and also the issue that I mentioned at the beginning about the vaccine efficacy in subgroups. And again, I don't think any of these is a perfect approach, but together they may give us enough information to make more evidence-based decisions. So for the infectiousness and herd effects, one approach that's being used in at least the Oxford trial of the AstraZeneca vaccine is to follow trial participants for subclinical infection. So that's sort of the gray bar that I described in my one of my earlier slides of if we could have perfect knowledge of when people get infected. You can do that for enough money and enough testing if you have a compliant enough population, and they're trying to do that in Oxford. You could add on households or other contact groups to vaccine trials. You could perform fully cluster randomized trials. And all of this is best done during a period of early scarcity when randomization is practical and clearly ethical. As we move through the approval process, this may become more challenging, although I think still feasible. And then for the vaccine efficacy in particular subgroups, I think one option is to combine data from multiple trials where they exist. Another is to continue trials beyond the emergency use authorization or whatever the first use case is. And I think that may be feasible, especially if the early emergency authorizations or equivalents in other countries are limited to certain subpopulations, as they may be. We heard a lot about correlates of protection, and that may help us to evaluate at least likely vaccine efficacy in subgroups. And then observational studies building on the experience with flu, where you look at people who chose not to get the vaccine and compare their frequency of getting the infection is another possibility. The last approach that I want to mention is to just ask the question, maybe actually this scientific knowledge is of course important, but not essential. Maybe there are some conclusions we can draw about how to use a vaccine that are robust and don't depend as much on vaccine characteristics. And this is also a preprint with these collaborators. Kate Bubar, the graduate student at University of Colorado with Dan Larimore, led this work. And the slides, most of the slides in this part are slides that Dan made, which means that they are better than the ones that I've made. So again, the approach we've used is to simulate epidemics with people moving between these compartments in an SEIR model. But compared to a simple model like the ones that we used for the previous work, we need a little bit more detail. So in particular, it's been shown in a number of studies around the world that people's social contacts depend enormously on their age, and people tend to mix a lot with people of their own age. You can see parent and child mixing in the sort of wings or off diagonal here. And these patterns vary quite a bit between country. And these studies are now have been repeated in almost every country except the United States, oddly enough. But for many of the countries in the world, we have some data on social mixing patterns. So the idea of this work was to say the vaccine is likely to be scarce. We don't know a number of important questions, including whether it will work very well in the elderly, whether it will be transmission blocking. But what we can do is to vary both these unknowns and some of these some of these demographic and age contact variables and ask the question, do we get the same recommendations for prioritizing that we would under a wide variety of assumptions, or is it very sensitive to what we assume? And the assumptions that we are, or the question that we ask that I'll talk about in the next couple of minutes is about prioritizing for mortality. The preprint has information on what to do for infections. And I'll just say that question, if you're trying to minimize infections, there is more sensitivity to the details. But for mortality, it turns out that what I'm going to say on this slide is true no matter almost how you tweak the model. So what we compare here is prioritizing in one particular country. I believe this slide is from the Belgian data, prioritizing adults over 60 to get the first vaccines, adults over 20, all ages, adults 20 to 49, and young people. And if you are interested in minimizing mortality, then giving the first limited supplies of vaccine to adults over 60 does so more effectively than any other groups for a simple reason of direct protection. These are the people at most risk and of dying if they get infected, and they are at comparable risk to others of getting infected. And so it follows that vaccinating them is the most effective for mortality. Doesn't matter very much what the efficacy of the vaccine is. And I'll just note that it's a different prioritization from who you would vaccinate to minimize infections. We know that many vaccines, for example, the flu vaccine, become less effective as people age. And so we considered, for example, a setting where the vaccine is 100% effective until 60, till age 60, but then declines to being 50% effective in 90-year-olds in a stepwise fashion. And what you see is that, of course, that doesn't help. That makes it worse. But it doesn't change the order of prioritization. It's still better to vaccinate the elderly first and everyone else thereafter. And that's robust when we vary the time at which, the age at which vaccine efficacy begins to decline, and the baseline efficacy and how much vaccine you have. So all of these things where there's a dash say, under all circumstances, even if it's 0% effective in the 90-year-olds, it's still best to start with the elderly, because most people are going to get, say, a 10 or 20% benefit, and their risk is so much higher that it's worth it. It's also robust to whether the vaccine protects against transmission and infection or only against symptoms and death, again, for the same reasons. And it's also robust to demography. So if you compare different countries, you get the same results. And it's also robust to whether the vaccine has a leaky mode of action or an all or nothing mode of action, meaning that some people are completely protected and others are not protected at all. So to summarize, when we have to roll out a vaccine, we're going to have imperfect information about efficacy in high impact subgroups such as the elderly and comorbid, and the impact on infection and transmission. There are approaches such as post-trial serology or regular viral testing of all participants to help estimate the impact on transmission. We can potentially use viral sequencing during the trial to provide an estimate of infectiousness impact. There are several preconditions for that to work, and I'm not asserting that those are all met for COVID-19 trials as they're currently designed. But one good bit of good news is that the basic principle of prioritizing the elderly or comorbid, if you want to minimize deaths, it's robust to many different vaccine types, and won't be far off optimal, almost no matter what we find about the vaccine that's plausible. We have not considered prioritizing essential or healthcare workers, which are also serious considerations. So this is just looking at the question of age. And we still need to know these properties of vaccines, even though we could prioritize relatively effectively. Even if we don't know, there are some questions that really do require knowing. For example, if there's herd immunity, we need fewer doses. If we were trying to block transmission, say by immunizing staff in nursing homes, is that going to work or is that not going to work? So there are many things we need to know about these properties of the vaccines, and I'm not saying we can just skip that, but I am saying that that fortunately it's not exquisitely dependent, at least which ages should go first, because the results are sort of similar, or quite similar, almost regardless of assumptions. So I'll stop with questions, and I'll just refer to this textbook, which for those who are statisticians and like to read a statistics level treatment of vaccine trials, this is the book that I give all my students to read, and it's the state of the art as of a few years ago on which we have tried to build some additional work.
Thank you so much, Mark. It's great to have more of a sort of a public health perspective on these questions. So we really appreciate you providing that in this meetup. We are past time, but I would like to go to at least a few of the questions. We won't go too long. And the question that has come up a number of times and certainly is on a lot of people's mind is everyone knows that typical vaccine development takes a long time. We've seen a lot of numbers, but 5 to 10 years is probably a conservative estimate. And everybody knows that pharmaceutical companies and the rest of the world are really trying to condense this. And there's always the question then of, are we sacrificing quality, and what are the parts that we're really being able to accelerate? And so I'd like to hear some comments on, what are the most important parts to accelerate, and how concerned should we be about whether quality is being sacrificed? So I know, Ivan, you had a slide on that, and Josh as well, but any of the panelists who'd like to take a crack at that, we'd love to hear your answers.
Hey, Dan, maybe I can take the first shot at it. I think if you see that a lot of the criteria that the regulatory agency put down is still very rigorous, high bar in terms of efficacy and safety. Some of this acceleration really comes around taking more risk in preparing for manufacturing, right? We're trying to run a phase 1 and jump over to phase 3, but the phase 2 is still ongoing. So you're taking a risk. And then if we find out there's a safety signal in phase 3, I think then the risk on the manufacturing sponsors, right? And for example, they also started the manufacturing of the vaccine while phase 3 are ongoing, but that doesn't mean that we're gonna release the vaccine before we have the formal approval. But if the vaccine goes down during phase 3, then all the money and investment lost. So I think it is a lot of acceleration is really the preparational work and the resource that's spent. But I think the good thing is that we are still holding that quality criteria from a safety and efficacy point of view, as far as I'm seeing it now.
Great, thank you, Ivan.
So just want to add to what Ivan mentioned. So you see this time saving here most happen actually by going faster. Now think about the research part, right? Typical vaccine from research, from the bench, to animal testing, tox study, to clinical, usually take 3 years. Now here we're talking about just a month, just a few months, because a lot of things can happen. The genome of the virus came out, as Ivan showed in the slide. It's just a matter of days, and mRNA came out just a few weeks. Can come out with the designed mRNA, and also some of the platforms already established, so you don't have to go through all the preclinical animal study, tox studies. So that's where you can save. And also in clinical space, you have this overlap, phase 1, phase 2. That's truly accelerate. And when manufacturers make the clinical lot, usually sometimes they have to need several months, maybe years to make phase 2 and phase 3 materials. But here they actually can make, as I mentioned, they start making clinical material at the time they start preclinical assessment. So all this actually squeeze in overlapping phase and going faster, and that can save a lot of time.
Great, thank you. And Mark, from a public health perspective, are we missing things by going so fast?
Yeah, I have a few thoughts. I think one thing to say is that after Ebola, there was the establishment of CEPI, the Coalition for Epidemic Preparedness Innovations, and some other efforts to to work on coronavirus vaccines. Not for this one because it wasn't known about. And I think we shouldn't forget that part of the speed is possible because people were working on very closely related vaccines already, and so some of it is actually public health doing exactly what it should. That Jeremy Farrar and Stanley Plotkin and Adele Mahmoud wrote their op-ed in the New England Journal, and CEPI was soon born, and that was in 2015. So I think it's actually, there is a success story there that shouldn't be forgotten, of learning lessons from earlier epidemics. The second thing to say, I think, is I agree with all of what's been said. I think the risk is greatest when politics and science get mixed, or when politics begins to infiltrate science. And I admire the statements from the vaccine companies that they've stated they will not cut corners. But I think that that in order to get out of this pandemic, our standards might not have to be, I mean, speaking personally, from my opinion, I don't know that the vaccine, in my opinion, needs to be as safe as a vaccine that's not going to get us out of a pandemic. I would be willing to tolerate some sore arms. I'm not talking about severe adverse events. A lot of sore arms if they could get us out of the pandemic faster. So I think there's a social conversation that needs to be had about what level of safety is acceptable. But I think the critical thing is to keep it public, transparent, and very clear what the trade-offs are, and ensure that the safety concerns are met, and that efficacy is not just a whiff of a hint, but is really evidence.
Okay, great, thank you. And so while I have you, let me stick with you, because I think this question was directed to you, and I know you've thought about this. The question was asked, what about human challenge trials? Is that something that we're likely to see in the future? And what are your thoughts on that as a step forward?
I think at the moment we wrote an article in March proposing that such trials could be ethical as a way to accelerate vaccine approval. At the moment, there is some effort to at least prepare for such trials in Oxford and at the NIH, not to do them, but to prepare for them. And I think there are still some circumstances under which they could be very valuable, especially if the first round of vaccines doesn't turn out to be sufficient to meet global needs for whatever reason. Having said that, because of public health failures in some very large industrialized countries, like the United States and Brazil, and to some extent the UK, there is enough ongoing transmission to run phase 3 trials. We had hoped that would be not the case, and that we would have pristine populations waiting for a vaccine so that they could let up on the control measures. But that's not how things have evolved. And so while I continue to believe that it's ethical if needed, it's not clear what exactly those will be needed for. But I think we will see as things evolve whether the phase 3s now in progress and starting in the future will be enough, or whether challenge trials have a role to play.
Great.
So if I can add just a couple of thoughts. Human challenges study, I think it is, as Mark mentioned, if the pandemic epidemiology changed, right? Because it's very difficult to predict. If it happens, could be one innovative way to address no cases anymore. So that's the way to look at it. And the ethical part, because the therapeutics actually is advancing. There are potentially, if we have therapeutics available, and we can identify the infection very early on. So that way I can address the ethical concern there. So could be, I agree with Mark, this potentially human challenge study could be a very powerful tool down the road.
Great, thank you. Going back a little more to the pharmaceutical side of things, there are a number of questions about the regulatory framework and the emergency use type way forward. What do you guys think about, is that going to be adequate? Are there changes that you think need to be made with that? Or can pharmaceutical companies work within the regulatory framework that has been put forward?
So if I can take, so the EUA plus, that is actually a new concept, just came out last week. And in the coming weeks, there will be additional guidance on what exactly the EUA plus means. The idea is to separate that from the EUA, which has been used in the past a few weeks, a few months, for some therapeutics. The idea is EUA that it's going to be, my understanding, like for example, mentioned the interim analysis, you must have 80 cases, which is put on the table, like for example. So that's efficacy included. So that's higher bar than EUA. And also EUA plus, it's not as high as traditional approval because at that point you can just use the interim data. You don't have to have long-term efficacy. Like for example, you don't need to full protection up to several years. You don't need to have the manufacturing data, like a lot of consistency data. You don't need to have that for the EUA plus. So in a sense, EUA plus is something between the EUA traditional sense and the traditional paradigm, which require all the data to get approval.
So that was probably just for the, I guess, accelerated approval, and then you will still need to follow up with more like a longer term the clinical trial data to support the regular approval down the road. But having this EUA and EUA plus, I think is a really good step forward to bring the vaccines earlier to the people who really need it.
Great, thank you. I'm going to have one more question for you, Mark, then a question for the whole group, which you guys may or may not decide not to answer. But let's, Mark, someone asked, have you considered about prioritization in minority groups in the US? Is that something that you can look at with your models and something that should be looked at?
Yeah, I think it is something that can be looked at. We haven't looked at it yet. I think that there are a number of complexities there. And I'm not talking about the political complexities, which I think pull in both directions. But if it is unquestionable that minority groups have been hardest hit by this pandemic, both in terms of infection rates and conditional on infection rates, their risks of dying, for all sorts of bad reasons, and for many reasons we don't fully understand. One thing that we have learned from other work in the past, looking at flu and age groups, is that the groups that are most hard hit early on in an epidemic are then less hard hit later on in the epidemic because those subgroups have built up herd immunity. Now, there are a lot of questions about what herd immunity is in this infection, how people's mixing patterns work, and the like. But the general point is that it might be the case that in the later phases of this epidemic, as herd immunity has built up to some degree, if it is effective, that not only will individual members of minority groups who have been infected before be less at risk, but their contacts will also be less infectious to the extent that we have residential segregation and mixing, assortative mixing by race and ethnicity. So it's actually possible that the high-risk groups will change over time. For that reason, I think it's worth modeling, it's worth thinking about, and I think we probably will try to do that. I think it's really, really hard to come to scientifically robust conclusions. And for all the reasons I described before about sort of, we don't know all the things about the vaccine, I think this issue will become even more complicated when trying to prioritize by race ethnicity. But having said that, I think it's probably worth trying to do. It's just, I'm scared of the consequences in terms of trying to explain the scientific uncertainties and not get mired in politics.
Sure, it's a hard scientific problem and the politics make it harder. Okay, I'd like to pose one last question, and I do understand that nobody has a crystal ball, so you can give your opinions, but nobody's expecting a perfect response here. But the question on everybody's mind, of course, is when are we going to have a vaccine, a safe and effective vaccine? Most of us have very little experience with vaccine development, and so we're all left in the dark. We realize that you guys, just like the rest of the world, don't know. But just some comments on what you expect to unfold.
Yeah, if we look at the three phase 3 trials going on, two of them are mRNA vaccines, and they require 2 doses. And you track some of the enrollment of the phase 3 trial. I think it's likely they're going to complete the enrollment either by the end of this month or early October. But that's just the enrollment, and then meaning the first dose, and then you need to wait for another month to finish the second dose. And then I think one of the trial protocol I look at is that they start tracking the cases for primary analysis at 14 days afterwards. So that would take you to almost mid-November, right, to start have a full follow-up for all the people that have been enrolled. Of course, they're going to do the interim analysis, and I guess what Josh is saying, that there's a higher bar now, even based on the interim analysis, you need to wait until 80 cases. If we target for 150 and you need to wait 50 cases, there's that midway through the. So I would think probably the earliest would be maybe early next year. That would be my best guess.
Thank you, Ivan. And like I said, we will not hold you to that. Any comments from Josh or Mark?
So for me, I think there are 2 questions here. One is when we can have the early readout on the positive results, right? That's one question. As Ivan said, well, people are talking about interim analysis. The interim analysis, of course, you need to, how many cases you need. And if you only plan 32 cases end of October, which probably not sufficient enough to be accepted by some of the regulators, they probably want to see 80 cases. That's going to push all the way to maybe end of the year and early next year, as Ivan mentioned. Yeah. And of course, at that point is, will you have sufficient, the efficacy could be good enough? Because this point, we haven't seen any efficacy in humans. And that's going to be the second uncertainty. We don't know if we're going to see positive results. Now, the second question is, after we see the good results, what's going to happen to make vaccine available to people? And someone said even the best case scenario, by end of this year or October, we have the first vaccine approved. That's going to take 4 years to provide vaccines to people in the earth. So that's a lot of manufacturing to do. So the 2 questions together, I wouldn't think vaccine going to be the will solve everything. It's going to be continue again in the US. Only 70%, less than 70% of people say they're going to get the vaccine. No more than 70%. So it's not going to be sufficient enough for herd immunity. Still, social distancing, all these measures were still in place. So in that sense, in the next year, the year after, we probably won't go back to the normal life before COVID. Maybe I'm a little bit pessimistic, but
But again, you guys are speaking from experience. Mark, any comments you want to add to those?
Yeah, I mean, I think it really depends on who we are, right? I mean, the United States has decided to go its own way and try to get hold of supplies of many of the leading vaccine candidates. We don't know if those are going to be the winning ones. The United States has decided not to opt into COVAX, the global initiative to try to provide vaccines to those who need them most. So I think it depends on how you define we. But if we define we for the United States, I think I probably fall a little more into Ivan's optimism than into Josh's pessimism. But Josh is also talking about the globe, and Ivan was talking about the U.S. So I think that's the critical thing. And I also think, I mean, I really hope that the line is held on what constitutes a reasonable signal of efficacy and safety. My fear in looking at the politics of it is that the worse the data are, the less compelling the data are, the better a political fight it can be turned into. And the incentives are all wrong, and we've seen the politics get in the way. So I'm actually quite worried about that, at least in this country, but let's hope that I'm wrong.
Well, thank you for your comments. And I really do want to thank all three speakers. I think this was a tremendous service to the population at large, but mostly these are statisticians who are tuning in and other folks. You guys gave a great introduction, really touched on all the important issues and really, I think, enlightened everyone in terms of what's going on. And you all know that this is a pretty important topic for just about everybody. So I want to thank all three of you for volunteering your time, for staying overtime. I want to thank our audience, obviously, for attending. And I certainly want to thank the folks at NISS, especially Jim Rosenberger and Glenn Johnson, who helped technically put this together. So with that, we'll sign off. As I mentioned before, all the slides and recording will be at www.niss.org. And I hope everyone has a good rest of the week and stay safe during the pandemic. So with that, thank you, Vince.